Hi, hello, and welcome to Dr. Vinita's bio classes. The aim of this channel is to develop an interest in students towards biology. We are starting with Unit 6, Reproduction. Before each unit, you are being acquainted to a scientist who has played a major role in science, in life sciences mainly, whether it be in field of botany, zoology, or any specialized fields of life sciences. Dr. Panchanan Maheshwari was a botanist or a phytologist. What do you mean by botanist or phytologist? Any person who studies about plants is known as a botanist or a phytologist. So he was from Rajasthan. He was the first person to introduce a technology, or he actually invented the technique of test tube fertilization of flowering plants. That is angiosperms. Angiosperms is flowering plants. And this helped in the development of new hybrid varieties of flowering plants which would not have crossbred naturally. So this technique of test tube fertilization was invented by Panchanan Maheshwari, and he is known as the father of Indian plant embryology. He is also responsible for this very textbook of NCERT on biology in the year 1964. So that is about Panchanan Maheshwari. So we'll quickly move on to chapter 1. So have you heard about the term floriculture? Floriculture. It is the cultivation and management of flowering plants. We all know flowers as a symbol of aesthetic beauty, right? But for biologists or botanists, it is the site for sexual reproduction. So there is a homework given in your textbook to find out 5 plants which can be cultivated at home and 5 other plants which are used in social or cultural celebrations. So you can just list down 5 each in your notebook. Okay. And one thing I would like to tell you is that you should be having your textbook with you, and you can have a scribbling notebook also, a rough book also with you, in which you can take down the notes, or you can underline the points that I tell you which are important in your textbook itself. So keep your textbook along because we are following each and every sentence written in your textbook. Okay, so now we are moving on to the first heading in the textbook, pre-fertilization, structures and events. Pre-fertilization. What does this term pre-fertilization mean? Do you know about fertilization? Fertilization is the combination or fusion of the male and the female gamete. So any process which comes before this fertilization is known as pre-fertilization events. So in this part we will be discussing about the pre-fertilization structures and events. Okay. So have you seen a bud? A bud will be an undifferentiated group of cells, right? It will be a homogeneous group of cells. Homogeneous means what? Homogeneous. Homogeneous means homo means same. So it will be a same, like all the cells will be exactly the same. It is completely undifferentiated. When you see a bud, at the initial stage of a bud, you won't be able to differentiate the different parts of the flowers. You won't be able to appreciate the difference in any of the cells. It is a homogeneous group of cells. Okay, so this homogeneous group of cells is known as floral primordia. Okay, floral primordia. This floral primordia, it undergoes differentiation and development by the influence of several structural and hormonal changes. Okay, so several structural and hormonal changes happens within this flower or floral primordia, which results in its differentiation. And what is meant by differentiation? This is another term. Differentiation is actually the cells attaining different forms and functions. Okay, the cells get differentiated to attain different forms and functions. So the several hormonal and structural changes in the floral primordia results in the differentiation, and which results in the development of floral parts. Okay, that's how you can differentiate. That's how it happens. Like initially it will be a floral primordia which won't have any kind of differentiated cells in it. It will be a homogeneous group of cells, and this homogeneous group of cells undergo differentiation by certain hormonal changes in this floral primordia, and this results in the development of floral parts. Okay, and you might have also heard about a term called as inflorescence. Inflorescence means it's just that a cluster of flowers on a branch. Okay, there are certain flowers you might have noticed that it doesn't come as a single flower. There will be a group of flowers. It's a cluster of flowers. That is known as inflorescence. Okay, so flowers, they differentiate and undergo changes and develop the male and the female reproductive structures. The male reproductive structure is known as androecium, and the female reproductive structure is known as gynoecium. This androecium is a group of or a world of stamens. Okay, you'll be learning about its structure now. So androecium has a world of stamens, and gynoecium is the female reproductive structure. So now let's see about stamens, microsporangium, and pollen grains, which are all the pre-fertilization structures. First of all, let's see about stamens. The stamen has two parts. One is the long, slender stalk, which is known as the filament, and other is a bilobed structure. It's bilobed structure on the terminal end, which is called as anther. These two together is known as stamen. Okay, so stamen at its proximal end is attached to either the petal. In some flowers, the stamen will be attached to the petal, and in some others, it might be attached to the thalamus. What is thalamus? Thalamus is the hardened structure about the stem. The flower is usually attached to the stem part, and you'll see a thickened part at the end of the stem where the flower is attached to. That part is called as the thalamus. Okay, so this is how a stamen looks like, and it has a long stalk, which is called the filament, and a bilobed structure, which is known as the anther. These two together is known as stamen. Have you seen the stamen? Well, let me share you one of my childhood memories. We used to play with the stamens. Yeah, literally, we used to play a war with the stamens. And you know a plant, a flower called the bullmohar. The bullmohar tree it flowers during the summer season, and it has the inflorescence. Inflorescence I had mentioned before. It is a cluster of flowers on a branch. Okay, so this bullmohar has this inflorescence, and the flowers in the inflorescence has very prominent, long and very big stamens, and the anther are quite prominent that you can just hook up the both the anthers, two anthers, and just pull it. So that was the game we used to do. Both of the partners, the opponents would have a stamen with them, and they would hook up the anther and just pull it, and whose anther falls down loses the game. So that's how we used to play. And the stamens, the number and the size of the stamens and all will vary from species to species. Like some stamens will be smaller in size, some will be bigger, depends upon the flower size and all. Okay, so the anther is actually a bilobed structure, we told already. And this bilobed structure, the anther, if we take it out, like this is how it looks like. It's a bilobed structure. It has two lobes. If we take it and cut it over here like this, like we take a cross section of it, a cross section if we take, we'll know more about the details. A cross section, for example, you have a banana, so you cut it, that will be the cross section, right? You'll be cutting it through the middle, so that is the cross section. And another is like you are making a banana fry, you will be cutting it lengthwise, right? That is the longitudinal section. So two ways of, in biology, usually scientists use these two ways of studying the structure of a cell or a tissue. We already know that it's bilobed. When we cut it, cross cut it, we find that it is tetragonal, like it has four parts, like it is bilobed, and in those lobes you can again find thecas. Okay, so each lobe has a theca. Theca is nothing but a cavity. Okay, so each lobe has two theca in it. So similarly, so in an anther there will be four thecas. Okay, so it is bilobed and each lobe is dithecous. So total of four thecas. You saw it as two lobes, but there is another groove going through here. Okay, so that's why, because of that groove, when you take a cross section, you can find this four thecas. Okay, so that's how it looks like. And so now we can say that the anther is four-sided. So it can be called as tetragonal structure. Tetra means four, so it is a tetragonal structure, anther. And the anther has four microsporangia. Microsporangia, okay. Microsporangia. What is meant by microsporangia? What is meant by sporangia? Sporangia is sac-like structure, and microsporangia means microspore producing sac-like structure. Okay, it is a microspore producing structure. Okay, microspore producing structure, sac-like structure, which is known as microsporangia. So it has four microsporangia along the sides of these theca. Okay, these four sides of the theca, or the four corners of the lobes, it has the microsporangia. Okay, so in each lobe there will be two microsporangia, or you can say that four microsporangia in one anther. In each theca, like theca, there are four theca, right? Four thecas are there. Theca is cavity. So in these four theca, there will be microspores. Microsporangia, like sac-like structure, which produces the microspores. Okay, so we have the microsporangia in the anthers, four microsporangia in one anther. Okay, and this microsporangia, which is a sac-like structure, it develops into a pollen sac. A pollen sac which produces pollen grains. Okay, pollen grains is actually the male gamete. It is the stamen is the male reproductive structure which produces pollen grains which are the male gamete. Okay, so male gamete is produced inside this microsporangia, which is located in the theca of the bilobed anther. Okay, understood? So this microsporangia will develop into a pollen sac which will fill the whole of the anther. This whole of the anther will be filled with pollen grains. Got it? Okay. So next is the structure of microsporangia. So this was the structure of the stamen. Now next is microsporangia. Now is the structure of microsporangia. You can see in your textbook the cross section of the microsporangia, the cross section of the anther, and in the anther you can see the microsporangia. Detailed picture is shown in the next page. Okay, so as told, it's a tetragonal structure. Okay, the anther is a tetragonal structure, and if you look into the picture, the anther has, in its cross section, you can see 4 layers, right? The outer layer is the epidermis. Outer layer is the epidermis. Then comes the endothecium. And then comes the middle layers. So these 3 layers, like it has 4 layers, but the epidermis, endothecium, and the middle layers, these 3 have the function of protection. They protect the microsporangia or the pollen grains, and it helps in the release of these pollen grains by the dehiscence of the anther when it is matured. Okay, so this is actually a protective layer, and this epidermis, endothecium, and middle layer. And there is also a 4th wall which is called the tapetum. You can see the 4th layer called the tapetum. The tapetum is actually a dense cytoplasm. It has a very dense cytoplasm, and it has 1 or more number of nuclei in it. Okay, and the main function of tapetum is to give nutrition to the growing pollen grains or growing microspores. It gives nutrition to it. So that's why it has a very dense cytoplasm. But the number of nuclei can be 1 or more than that. How? You know that all the cells divide, right? All the cells do divide, that's a necessary thing. But if the tapetal cells also divide, then the space that will be required for the pollen grains to grow within, for the microspores to become the pollen grains and to become mature, it needs space, right? It's now when the anther is immature, the space is little, but when the when the anther grows, the microspores also has to grow and it has to become the pollen grains. They undergo only karyokinesis. What is meant by karyokinesis? Karyokinesis is the division of the nucleus. The nucleus cells, they divide, but the cytoplasm doesn't divide. So this type of division of the cells is known as endomitosis. Like mitosis happens, but it happens within only the nucleus. So nucleus divides, but the cytoplasm doesn't divide. Okay, so karyokinesis occurs, but the cytokinesis doesn't occur. The cytokinesis is the division of the cytoplasm, that doesn't occur. So that's why, that is the reason why the number of nucleus is will be more. The number of nucleus can be more in the tapetal cells. The main function of the tapetal cells is to give nutrition to the microsporangia or the microspores and it also has a protein called as sporopollenin. So what is meant by microsporogenesis? Only the new part which has been added to it is the genesis. Microspores we have already understood, right? So this microsporogenesis simply means the process of formation of microspores. Okay, the process of formation of microspores from the microsporangia in the microsporangia is known as microsporogenesis. Understood? Okay. So as the anther develops, the sporogenous tissues, which will be initially homogeneous in nature, the sporogenous tissue they undergo meiosis. Okay. So what is the difference between mitosis and meiosis? We have already studied, right? Mitosis is a process which happens only in the normal cells, but meiosis is a process which is exclusive for the sex cells. Okay, so the sporogenous tissue, they are the factories for the production of the male gametes, right? So these sporogenous tissues, they undergo meiosis. With mitosis, two daughter cells will be produced, right? With the normal mitosis cell division, two cells will be produced. But with meiosis, how many cells are produced? How many daughter cells are produced? 4 daughter cells are produced, right? With mitosis, only 2 will be produced, but with meiosis, 4 daughter cells will be produced, right? And what will be the ploidy? In a normal cell, there will be 2 sets of chromosomes, right? And in a sex cell, there will be only 1 set of chromosome. In the mitosis results in 2 daughter cells, which will be having 2 sets of chromosomes, and so it will be called as diploid. And in meiosis, there will be 4 daughter cells, and it will be haploid. Haploid, in the sense it will be having only 1 set of chromosomes. In this sporogenous tissue, they undergo meiosis, and 4 cells will be produced. 4 daughter cells will be produced. And so, these 4 cells are called as microspore tetrad. Like the sporogenous tissue, each individual sporogenous tissue, they undergo meiosis and 4 microspores will be produced, and so it is known as microspore tetrad. Okay, and each sporogenous tissue is a mother cell, pollen mother cell, PMC, and these pollen mother cells produce microspore tetrads. The process of formation of microspores from pollen mother cells by meiosis is called as microsporogenesis. So it's actually, the process is known as microsporogenesis. Okay, so once this microsporogenesis happens and these microspores are produced, they remain in the anther itself. Okay, they remain in the anther itself unless and until the anther becomes fully matured. And once the anther is fully matured, it starts to dehydrate. It dehydrates, and once dehydration happens, these pollen grains or the microspores, which are now fully developed pollen grains, they are dispersed into the environment. Okay, the outer covering of the anther, due to dehydration, it dries up and it opens up for the pollen grains to escape into the environment. Okay, that's how the pollen grains come to the outer world. So now about the pollen grains. So now we have seen about stamens, we have seen about microsporangia, and the third is pollen grains. Now we have to see about pollen grains. How does the pollen grain look like? We saw that about when in the cross section we had seen how microsporangia and things and all we have already seen. Now individual pollen grains, if we see, and see the hibiscus plant, you know that the hibiscus if you take a hibiscus flower and you just tap it, the flower on your fingers, you can see that even if you take the stamen also, you can just tap it and you will find yellowish powder, right? Those yellowish powder is the pollen grains of the plant. Okay, so how does the pollen grain look like? Pollen grains are generally it will be spherical. Usually it will be spherical, but there are exceptions too. Different shapes are also there, but generally, mostly it will be spherical. Okay, and the pollen grains, you cannot visualize the pollen grains through naked eye. You can just see it as dust. If you want to see it clearly, you have to see it under the microscope. You will see it as a ball. Okay, you won't be able to see the details of it. You cannot see the internal structure of the pollen grains, because pollen grains, the outer covering is quite thick that you cannot see into the inner sides of it. So, and you can also see it using electron microscope. So the difference between electron microscope and light microscope is that in the light microscope, the source of light is any lamp, tungsten lamp or any kind of lamp will be used, or laser can be used in the light microscope. But in an electron microscope, electrons are used. In your textbook, you can see that there are a few pictures of the pollen grains, and the middle picture is of the image taken in SEM. SEM is scanning electron microscope. Scanning electron microscope shows you the morphology, like the how a thing looks like. Any specimen that you keep under a SEM or scanning electron microscope, you can see the morphology of it in 3 dimension. Okay, so that is the image that you see in 3 dimension. You can see the structure of the of your sample or the pollen grains. So in that itself, you can see that there are several shapes of pollen grains in it, right? Okay, so if you want to see it under light microscope, then you will have to see it with a normal light microscope won't be enough. A plain polarized microscope can be used to visualize the internal structure to an extent. Okay, so usually the pollen grains are of the size 10 to 150 micrometers. Okay, in your textbook it is written as 10 to 20 or something, 10 to 50, some small range is given, but actually it is from 10 to 150 micrometers. It can be from 10 to 150 micrometers. Do you know how much is a micrometer? You know you know about a centimeter, right? Centimeter. In a scale, 15 centimeter scale, you know 1 centimeter. You know a millimeter. Millimeter is the 1/10 of the centimeter, right? There will be 10 divisions in the centimeter, and the smaller part is the millimeter. And micrometer is 1000 micrometer will make up to 1 millimeter. Got it? Like this 1 millimeter, the 1000th part of the millimeter is known as micrometer. So 1000 micrometer makes up a millimeter. Got it? So the pollen grains has, I'll just write it down, 1000 micrometer equal to 1 millimeter. Okay, this is the calculation structure of the pollen grains. So the pollen grains is almost, we said that it is almost kind of spherical. It has 2 prominent outer layers. If you draw pollen grain, it has 2 outer layers. Okay, so this outer layer, the very outer layer may be not that polished kind of a surface. This outer layer is known as exine. Okay, the outermost layer is known as exine. It is a very thick layer, and there is an inner layer which is known as the intine. Okay, this outer layer exine is made up of the protein sporopollenin. which I had already told you that the sporopollenin was produced by tapetal cells, right? Tapetal cells gives the sporopollenin to the pollen grains during its formation. So that sporopollenin is the most toughest of all the proteins till date. No chemical or temperature or any enzyme has been found out till date which can degrade this exine layer, the sporopollenin. So that is the sporopollenin is the hardest and toughest of any protein, and it can withstand any temperature, whether it be high or low. It can withstand any temperature, as well as it can it is not affected by any strong acid or alkali, and no enzyme can act on it. So that's the strength of this outer layer. But at some places there is a gap. Okay, it is not a continuous layer. Exine is it's not a continuous layer. At some places there is a gap. There are areas where this sporopollenin is not there, because that's very important. Because if this is that very tough an outer covering, then how would the gamete inside the pollen grain come out, right? So that's why there are certain places wherein there is no there is absence of this sporopollen. So other than that, there is intine, which is an internal covering. Okay, and that is made up of collagen and pectin. Collagen and pectin, they are all carbohydrates. They are collagen as well as pectin. They are structural carbohydrates. Okay, carbohydrates are of two types: storage as well as structural. So this is the intine is made up of collagen and pectin, and these two are the structural carbohydrates maintaining the structure of the pollen grains. And inside this there is another layer, the outer one, where both of them were called as, they are the cell walls. Okay, and internal to the intine is the cell membrane or the plasma membrane. Okay, plasma membrane or the cell membrane is another covering which is inside the two thick walls, the exine, intine, and then comes the plasma membrane. In 2013, few scientists found in Switzerland, they found nearly 240 million years old pollen grain. In Switzerland, they could find out. So like 240 million years old pollen grains means what? Like that was the Triassic era. Like Triassic era is the early dinosaur era. Like after the Triassic era came the Jurassic era. And so this pollen grain belonged to the Triassic era. So these scientists got the fossil of this pollen grain which was of 240 million years old. That is sporopollenin. Okay, mature pollen grains, they contain two cells. Okay, within this pollen grain itself, they undergo mitosis. You already know that the pollen grain is haploid in nature, right? The pollen grain is haploid in nature, and it has the sex. It is the sex cell, but inside the sex cell, it undergoes mitosis, and mostly this will be, and there will be a spindle kind of, like within the cytoplasm itself. Within the cytoplasm, there will be another cell. Okay, so this is one cell, and within the cytoplasm, another cell is there, and that will have a dense cytoplasm. This is the outer one, another cell. So two cells are there, but one cell, a spindle-shaped cell, floats within the cytoplasm of the other cell. So this is how usually it is. So this is the bigger cell is called the vegetative cell. Vegetative cell, and the spindle shaped smaller cell is called as the generative cell. Okay, generative cell. So within the pollen grain, they undergo mitosis and they form one vegetative cell and another generative cell. The vegetative cell is bigger in size and it has all the food reserve in it, and it has an irregular nucleus. It will also be large, but it will have an irregular nucleus. And the generative cells, it is smaller in size, it is spindle shaped, and it has a dense cytoplasm. The cytoplasm is very dense, and as well as the nucleus is also dense, and it floats within the cytoplasm of the other cell. Okay, so this is how the mature pollen grains look like. They will be mostly two-celled, and it is also been found that nearly 60% of the pollen grains will be two-celled, will be in the two-celled stage, but the rest of the 40% they have been seen to be in the three-celled form. It has been seen that the generative cell, again, this generative cell, the vegetative cell and the generative cell is there, right? And this generative cell, it will again undergo mitosis. Okay, so again this generative cell alone will again undergo mitosis, and that generative cell will be again two cells. So total 3 cells. So like that 3-cell forms are also seen in the pollen grains. Okay, so that's all about the pollen grains. And with regard to the pollen grains, you might be knowing that several people have allergy towards pollen grains, and they develop certain disorders related to the respiratory tract, like asthma, bronchitis, etc. And most of the cases, it is due to a plant, the pollen grains of a plant called as named as Parthenium. Okay, Parthenium is known as carrot grass. In Malayalam, we call it as Congress Pacha. Okay, so like that is a plant which is highly infamous of giving pollen grain allergy to people. And nowadays it has been seen that the pollen grains being highly rich, nutrient rich, several supplements of pollen grain supplements are also being given. People like athletes are given this pollen grain supplement tablets. Pollen grains, their viability is very important. Like the cells will be, the example I told you of 240 years old fossil thing, it had this outer covering, but the cell won't be there, right? The cell will be viable for maybe some might be viable only for a few minutes, and some may go up to a month or two. Okay, so that's the viability of the pollen grains. That is the cell, the gametic cells, the inside cell. We're not talking about the outside part. The outer part can live long, however long it wants, but the inner part, which has the vegetative and the generative cell, it has to be viable, and it has to reach its destination to release its gamete into the female reproductive organ, right? And that viability varies from plants to plants. In cereals such as in the paddy and the wheat, it has been seen that the pollen grains will be alive for only just 30 minutes. Okay, and some other plants, it may go even for months. So it depends from plants to plants. And another thing is that these pollen grains can be preserved. Pollen banks are available, pollen grain banks are available, wherein these pollen grains are preserved. And these are preserved in liquid nitrogen. You know about liquid nitrogen? The nitrogen exists in its liquid form at minus 196 degrees Celsius. Okay, and you can preserve things in this temperature in the liquid nitrogen. So the pollen bank is preserved in liquid nitrogen. Okay, so pollens can be stored so that it can be used after a very long time. It can be revived after a very long time. Okay, so that's about the second topic, that is stamen, microsporangia and pollen grains.